I'd like to talk about my work, which I call Venus Cycle. Umberto Maturana said a name is not just a name. Sometimes it's more than a name. And this is what I felt when I started in 1989. My works of art stem from an experienced deficit of our technicized everyday life. My installations build up their own value systems of ethics and aesthetics. This focuses on aspects which scientific operations exclude. This is Venus 1. As I said, I started in 1989, shortly after finishing my studies. Venus 1 is a work on how people acquire knowledge. It starts with Titus Lucretius Carus on the nature of things. At the beginning of his history of nature, he asks Venus to help him in understanding nature. He then describes the way he communicates with nature and helps Venus to help him. The last text in this project is knowledge that you have gained after sending a probe to Venus, the planet Venus. Venus is not the planet of love, it's the planet of heat. There's sulfur dioxide rain, it's a very dense atmosphere, a prototype of the greenhouse effect. Venus is a sister planet of Earth and can be a warning symbol as to what is going to happen if we continue to afford ourselves the greenhouse effect. In 1993, an article on CO2 in the oceans was the reason for an installation on the greenhouse effect, which is now in the Ludwig Museum in Germany. That was Venus 2. 18 years later, the federal government of Germany decides on the Carbon Capture and Storage Act. Another reason to resume this topic. This is my work from 1993, The Birth of Venus. In a wash basin, you can see plants in the background that absorb CO2. These are plants along the highways. And you have a text making the proposal to bury CO2 in the oceans. Then I have the greenhouse converter, which I call Venus 5, a natural cooperative model of CO2 increase. The installation symbolizes the processes along a human scale. This is a sensor that measures CO2 along the highways. The value has gone up to 358, and you can see that along the highway the value increases continuously. When you approach this fountain, surprisingly, it seems that CO2, or part of it, is removed from the air. The next thing I needed was plants that make sure that CO2 is converted in the water via photosynthesis. It's converted into oxygen and water. I started with experiments using algae that grow on matrices, and also they needed a partner eating these plants because you have to limit the growth of these plants, so I decided on plankton. And plankton eats up algae, which leads to a little cybernetic system. In the state beloved, both organisms, algae and planktons, are satisfied. In the state desolated, there's no harmony yet. That's the original state of the system. Red light means growth of algae. You can see that on the matrices. Algae move towards the matrix, towards desolated, the desolated state, and blue stands for beloved. That field or area remains empty because plankton are usually attracted by the blue light, so they don't eat up the algae. This is the first picture of the greenhouse converter. Beloved and desolated describe two states of a bio-cybernetic system focused on cooperation as the motor behind evolution. This is what the whole setup looks like. It starts with water. I didn't have the possibility to set it up outside near a road with a fountain, so I set it up in a museum, a small fountain. There's a user desk where people can pump water from the fountain into the aquarium and back again, so it won't flow over. And then there's also a picture for the user. He can zap through time. When you're in a museum or an exhibition and there are multimedia works, visitors expect things to change quickly, but this is something nature cannot do. These processes take a long time, and you need more patience than the average visitor will show. So this is why here you can zap through the history of this cybernetic system. So that was the first model of the greenhouse converter. Here you can see the plankton. The matrices are used as sensors and also to provide light. They can be used as sensors and as effectors. And outside the aquarium, there's a sensor which measures the light intensity and the color of light and compares it with the inside. So when algae have grown, it switches over to blue to attract the plankton. And there are also intermediate states between desolate and beloved. Here, the visitor can pump the water. If there's too little water in the aquarium, there's an indication, and the visitor can add water. Here you can see the attraction that blue water has on plankton. Here the original state, desolate. Next, I'd like to show you a project that I have developed with my students at the University of Heidelberg and the Cancer Research Center and Joland Eist. Last year, we set up the iGEM team. The project is carried out in Boston. We were thinking about creating a fictional supermarket, which you can find online. For a supermarket for speculative products of synthetic biology. I'll show you 5 minutes of the presentation of my students at the MIT in Boston. I think they provide a better explanation than I could give you.
Jetzt brauchen wir Ton.
Welcome to Supercell. Ladies and gentlemen, in a few moments, you will be witness of a great moment. Supercell is going to be opened up. The first supermarket which offers exclusively products that are based on synthetic biology.
A great scientific advance.
And a great moment for people all over the world.
Well, you may ask, why a supermarket?
Our project aims to improve the public awareness on synthetic biology and at the same time foster a debate on how we want to see it manifested in our everyday lives. Implications of biotechnologies are usually discussed in a philosophical, ethical, legal, or possibly religious level. All these areas share a pretty abstract perspective on the matter.
We design speculative products that are presented in a familiar environment, a web shop. By this, we aim to create an environment and a virtual manifestation of the vision we share for synbio products of the future. In this environment, specialists and non-specialists alike are invited to discuss the desired as well as the undesired implications of synthetic biology.
If we, as a society, wants to influence how this powerful science will affect our future life, this kind of discussion is essentially to have now.
Ladies and gentlemen, and here they come. This is the first public review. You will be the first to know what Supercell sells. What will be inside these boxes? Let's have a look on one of the exclusive products. Rico Brain, get your life organized.
Let's face it, there's simply not enough hours in the day to fit it all in. Multitasking just doesn't work, and there's not enough downtime. Thankfully, the people of Rico Labs have finally come up with an answer. Rico Brain is the latest scientific enhancement that allows for you to get fit, get organized, get ahead of your chores, all while you are sound asleep. Simply plug in the music and doze off. Think of all the time you can save with this ultimate form of multitasking. Rico Brain allows you to change time from what you have to do to what you want to do with just one minor procedure. Rico Brain is just one simple injection that allows for a carefree, happier, healthier you. But just don't take my word for it.
Thank you, Rico Brain. Now I finally have time to be outside.
Thank you, Rico Brain. Now I can spend more quality time with my family.
Thank you, Rico Brain, for giving me more time to relax.
The Rico Brain technology is based on the injection of a synthetic neuronal network that attaches to the brain's motor cortex and auditory cortex and induces the formation of new synapses. Neuronal activity patterns in these brain areas were recorded by the Rico Brain network and can be linked simply by listening to a specific personalized melody while asleep. Rico Brain is activated and repeats the corresponding activity patterns of the brain, causing the body to react to these stimuli by executing the programmed task.
Rico Brain by Rico Labs, exclusively at supercell.org. And
Ich zapp jetzt mal weiter.
Well, I can only recommend you to have a look at the film. You can find it on my website. This gave you an idea that was developed by Philipp Bayer and Martin Schumacher, two scientists, young students of biotechnology. And it was implemented in a cooperation with us. I myself pursued another idea using insects, and I wanted to teach them the letters, the alphabet. Chironomidae, these are insects, respond to frequencies. So we use a sound spatialization system in order to teach the insects. And they fly in swarms of simple LED letters. So you can actually train the insects with the proper arrangement of the loudspeakers and hopefully make them recognize letters. They form swarms in the form of spheres. The swarms consist of males who gather for mating. And we want to animate insects. I wanted to show you the video. Here we go. They didn't manage at first. They could hear the sounds, but they didn't respond. But after several training sessions, they actually gather in the shape of a letter. It's amazing that the insects can learn these things because they fly in swarms. And because they communicate in swarms, they can help each other in communicating a specific message. This is quite extraordinary. So these insects can truly communicate the message to their neighbors via the genes, via the exprimation of different genes. So we expect that we will be able to have them transmit a coded message. In practice, there were a number of surprises. Have a look. There were commercial messages that lost their shape. So it seems that, well, the message wasn't passed on to the next generation. Now, you should be seeing a lantern, yes, a street light, which of course disturbs the insect swarms. They don't know whether they should listen to the sounds or should pay attention to the light which attracts them. So what are the conclusions from these works? What was our experience with MIT last year and synthetic biology? We found that engineers of synthetic biology develop symbiosis, better animals, and we derive our self-confidence from the technological structure. We've designed this technology ourselves, but it has an impact on our culture with its artifacts. Art is effective in museums, galleries, art fairs, collections, and in people's homes. It's a leisure time activity for most people, separate from their working process. So who designs the society of tomorrow? Are engineers aware that they do not only design technical artifacts, but also our culture? Now, whether artist or scientist, we are involved in unconscious processes, breathing, metabolism, cell division, which we are not aware of and which we cannot perceive completely. As an artist, I rely on instinct, and I invent systems in order to overcome deficits, bodily deficits. in order to make use of man's full potential, we need an integration of this drifting in science and culture. There are people who have no access to the scientific apparatus. With every sensor that I use, part of the information that I use as an artist for my works gets lost. So my statement is, I'm a wild type. I don't have a genetic mutation, most likely. I don't have any bodily enhancements. I'm not looking for them. Rather, what I'm trying to do is observe my conditions and to create models for understanding. And these are models which I would like to discuss and develop further. And I would like to compare my models with models developed by scientists. Thank you very much.
Thank you, Ursula, and also thank you, Ariane. People who had to leave, but if there are some very burning questions, maybe we can still entertain them for our three speakers of this last session. My first thing is that perhaps Ursula should make presentations to Ariane, because it sounds one of the closest approximation I've seen between a really artistic sensibility and a truly scientific one. So questions from there is one right there. Can you say for who you want to ask to?
Sorry?
You would like to say who you want to ask the question to?
Yes, yes. I don't know if it's working. Is it working? Okay.
Yeah.
My question is, thinking on the relationship between art and science and how art and science can collaborate, there is one difference in the approach art deals with technology. Right? My question is for Ariane and for everybody, but it's especially for Ariane because artists want to torture technology. They need to torture somehow, or not always, but they have this possibility. And science is more, technology is more like a sacred thing. Thinking of the collider, if you are integrating artists in the CERN, how open are you to really allow artists to torture a little bit the machine in order not to be just illustrators or inspired by the CERN in a superficial level, but really to go into the machine in the sense that Flusser said, to go into the black box?
Art for me is absolutely about interrogation. It's about daring to do the impossible with the possible. So yeah, absolutely. When I say inspiration, that's only, it's like the science and technology of CERN is a springboard, and then it's up to the artists to do anything they like from that, interrogate it, revolutionize it, whatever. For me, that's the great thing about artistic freedom. It's about the imagination and liberation. So absolutely, this is all about the artistic process, the creative process, and then connecting it very much with the scientists. I didn't explain that very well in my talk. In fact, I come from the artist's point of view, which is quite unusual in an art science context. So yeah.
So they will be able also to provoke, to experiment with the machine, not only to be inspired.
Not use the machine. No, not use the machine because it's running.
The real collaboration between art and science, and when you give the tools to artists, and so they can really go beyond the inspiration.
Yeah.
My point.
Yeah. But if only we could hand over the machine to everybody, but I'm not allowed near the machine. Only one person is. But when it's running. So, but hey, who knows? You know.
Thank you. Another question? You're hungry? I understand. So have a lovely lunch. Thank you very much for attending this very interesting session. And I want to particularly thank Ariane Cook, Ursula Damm, and Roger Malina for their very exciting presentation. See you this afternoon.